Okay, so I was saying the bonding is basically due to the functional groups. When you're looking at hydrogen, you remember you're forming hydrogen bonds, and there's you're looking at a donor, a hydrogen bond donor, and an acceptor. So when you look at adenine and thymine, you have a donor and an acceptor. When you look at guanine and cytosine, they're going to make that a base pair because you're looking at the available hydrogen bond donors and the available hydrogen bond acceptors, which are going to pair together. So that leads to that hydrogen bonds, 3 hydrogen bonds being formed in guanine and uracil, and 2 hydrogen bonds forming in adenine and thymine. So you have hydrogen acceptor and a donor. So when you look at RNA, when you look at RNA, which is a ribonucleic acid, it is synthesized based on the information that is carried by DNA. And then, just a minute. There are basically 3 types of RNA. We have messenger RNA, transfer RNA, and ribosomal RNA. So from what we've looked so far, we know that RNA will contain nitrogenous bases of adenine, guanine, cytosine, and uracil in place of thymine. Therefore, for example, if you are measuring the synthesis of DNA, what you're going to use is a radioactive thymine or thymidine. So there are experiments that scientists can use. If there's DNA synthesis, they're going to measure the rate of DNA synthesis or RNA synthesis. So what you're going to use in DNA is thymidine, because when we use thymidine, we are sure that we are actually measuring DNA because it is not found in RNA. So when you look at a messenger RNA, simply it's a single-stranded molecule that is formed on a single strand of DNA by transcription, as we are going to be looking at transcription later or on another day. So messenger RNA is the one which contains the genetic code, which is according to the template at DNA from which it was formed. And then we have ribosomal RNA, which is normally a single-stranded nucleic acid synthesized from DNA located in the nuclear organize of several chromosomes. So it combines with protein to form the ribosomes, which are the sites for protein synthesis. Each subunit of each ribosome is made up of 2 subunits. So you have a larger subunit and a smaller subunit. So you have prokaryotic ribosomes, which is simply the ribosome that is found in prokaryotes or single-celled organisms, which is 70S. So you have the larger subunit of 70 or of 50, and then you have the smaller subunit of 30. So you have 50S plus 30S, giving us 70S. So the 70S, it's simply a sedimentation rate. So an experiment was carried out, a sedimentation experiment was carried out, which gave us 70. So it's not an additive or mathematical addition. It's a sedimentation rate given in Svedin units. That's the S, so it's a unit. Then you have the eukaryotic ribosome, which has a size of 80S. So you have 60S plus 40S sedimentation rate in Svedin units, giving us 80. Then you have transfer RNA, which is an adapter molecule involved during the process of translation. It is a clover leaf in shape, and it has 4 arms, which is the acceptor arm, the variable arm, the D arm, and the anticodon arm. Of importance to note is that the 3' end of the transfer RNA always ends in the base sequence of CCA. So this is simply a representation showing transfer RNA. So you have the variable arm, the anticodon arm there down, the D arm, the amino acid acceptor arm, and the T arm. So the amino acid acceptor arm there have the 3' end, which ends in CCA. The one we've mentioned, CCA, then you have the hydroxyl group there. So this is still a same representation of transfer RNA, amino acid accepting end there at the 3' end. Then comparing and contrasting. So if we are asked to compare and contrast between things, we are simply asking us to write the similarities and the differences. So similarities between DNA and RNA. From what we've looked at, the first thing we looked at was the definition of nucleic acid being genetic materials. So both DNA and RNA are actually nucleic acid or genetic materials. And then we looked at the formation of DNA and RNA. So we've learned that they're actually biological nucleotides. They are polynucleotides. So they are made up of repeating units of nucleotides. And then they are made of polymers, or they are made by the reaction of polymerization. So they are nucleotide polymers. And then they are present in both prokaryotes and eukaryotes. Since they are made of nucleotides, they both contain a pentose sugar, a nucleotide nitrogenous base, and also a phosphate backbone. Then in both DNA and RNA, we have adjacent nucleotides that are connected by the 3' to 5' phosphodiester bonds. And then both DNA and RNA are cleaved by nucleases. So these are enzymes that are able to cleave or degrade the nucleic acids. So in both DNA and RNA, we have adenine, cytosine, and guanine as nitrogenous bases. And then we have nitrogen hydrogen bonding in both molecules. On the differences between RNA and DNA, we have DNA, which is simply more of a storage material. So it stores and transmits genetic information, while RNA is usually simply transmits genetic information. When we look at DNA, DNA is mainly double-stranded, while RNA is mainly single-stranded. Then DNA has thymine in the place of uracil, while RNA has uracil in place of thymine. When we look at DNA, DNA is a huge molecule. It's a large molecule in relation to the relative smaller size of RNA. Or also, usually DNA is of one type, while RNA is many of 3 types. DNA or RNA is chemically unstable, while DNA is chemically stable. The reason of it chemically stable, DNA being chemically stable, could be explained by the 2' position. Remember of the ribose sugar, that 2' position of ribose sugar. We do not have the hydroxyl group in DNA at the 2' position. So the presence of that hydroxyl group, the OH group, and RNA at the 2' position makes RNA to be more reactive and unstable than DNA. Okay, then when we look at DNA in the nucleus, it exists not as a pure molecule, it exists in connection with proteins, or what are called histones. So that's one other information that we need to note. It is in association with histones, molecules in the DNA or in the nucleus. It's in association with histones. Okay, do we have any questions? Any questions?